Hello, thank you for joining me today for this Reaxys Medicinal Chemistry webinar. Today's topic is drug repositioning. Please send me your questions through the question box in your control panel or send them to me directly at c.fleming@elsevier.com. Drug repositioning or repurposing is a topic that has been gaining in popularity in recent years. I guess you could say that the reasons for this are obvious, considering the amount of time and the amount of money that it takes to get a drug to market the traditional way. Repurposed drugs have fewer safety concerns since marketed drugs and late-stage drugs have already gone through most of the safety hurdles. Repurposed drugs have a clear advantage. Many drugs that fail in the preclinical phase fail because they were not shown to be effective and not due to safety concerns. The large infrastructure needed for traditional drug discovery is not needed for repurposing, and the time it takes to get a drug to market can be reduced by several years. It also creates new opportunities in the area of neglected or orphan diseases. There are many approaches to drug repositioning. Mining databases is one of them. And that brings us to Reaxys and Reaxys Medicinal Chemistry, two databases with a similar data structure that can be searched together. Reaxys searches over 50 million unique substances, physical properties, spectral data, and reaction information. Reaxys Medicinal Chemistry contains over 5 million substances, 25 million biological data points from over 5,000 journals, and over 90,000 patents. And these databases can be searched simultaneously through one user interface. Reaxys Medicinal Chemistry is all about the connections between the bioactivity of compounds and their targets, and helps you choose the right compound to develop during your preclinical work. RMC gives you access to millions of compounds so that you can explore the pharmacological effect of selected compounds and investigate the targets with which the compound series interacts. And this can be used for the assessment of new drugs, lead identification, lead optimization, and, of course, drug repurposing. Our first example looks at a drug called ketamine. Ketamine's been used as an anesthetic for the past 50 years. Over the past 5 years, there's been some increased interest in studying its use in mood disorders as a vasodilator and an anti-inflammatory. Let's use Reaxys Medicinal Chemistry to get a profile of ketamine and its related drugs. Here's the interface for the combined databases. I'll begin by selecting the bioactivities theme, and then generating the structure for ketamine, and then run the search. The results open to the heat map, where I can view the data several different ways. Right now we're looking at ketamine and related drugs along the side, and targets along the top. And there seems to be a good variety of targets. Here's the sigma receptor, and here are the ionotropic glutamate receptors. What kind of effects have been observed with this drug? A quick look at the analysis tool shows you a listing, like behavioral symptoms, vasodilator, and even antifungal. I'd like to show you what the raw data looks like, so I'll open the substances view and click bioactivities for the first substance. Notice the details in the data, like parameters, cell line, target names, and bioassay names. You can export this data in a variety of formats by clicking the Output button. Here are examples of an exported PDF and an exported Excel sheet. In your study of ketamine, you may want to view substances that look similar to ketamine. Perhaps viewing their targets will suggest other possible uses for ketamine. I'll create the query using a similarity search. There are options for choosing the degree of similarity. The numbers are the number of bioactivity data points, not the number of substances. Here I can see the targets for ketamine and a few of its associated substances, and I can easily see the targets associated with the other similar structures. I'll click here to make the structures visible. Ketamine and the NMDA target are well studied, and ketamine is regarded as an NMDA antagonist. Find out more about this target by viewing data on other substances that bind to this target. Can I view only those that act as agonists? Set up the query using these two fields, target name and putative action on target. Then I'll choose the target and select agonist for putative action on target. In the results, I'll set it to display substances and effects. Then, to see which ones are most potent, I'll sort the list in descending order. Now with a list of NMDA agonists, I can view the data. For example, here's some information on the structure-activity relationship of NMDA agonists and antagonists. In the next example, I'm interested in finding a list of substances on the market that have been tested for antiparasitic or antiprotozoal activity. Reaxys Medicinal Chemistry has a field called Highest Clinical Phase. Here are the options. It shows the different phases, but also has options for substances that were discontinued during a certain phase. This may be interesting because some of the safety studies may have already been done. But I'll select Marketed for this one. And I'll select the effect, antiparasitic or antiprotozoal. Some of the substances in this list are drugs marketed for a use other than antiparasitic or antiprotozoal. For example, this one is a vasodilator. This one is used as an antioxidant. This one is used as a cough suppressant. And this one is used as an antihypertensive. Which targets are responsible for the antiparasitic, antiprotozoal activity? Just change the view in the heat map to effects and targets. Here are the targets and their effects. View the data by clicking on a px value. This one involves studies with caffeine and shows that the purine ring may serve as a useful scaffold in the development of inhibitors of trypanosomal CTP synthase. With many antiparasitics and antiprotozoals, the targets may not be known, but the biological species that they were tested on is known. Which substances had the highest px values? What biological species were they tested on? Change the view to biological species and then filter out the lower performing substances. Limit to rows you are interested in. For example, here's one used in eye drops for eye infections, and one used for cutaneous T-cell lymphoma. And here's a natural product used for a variety of illnesses. So here's your list of some products on the market with activity against Leishmania, Plasmodium, and Trypanosoma. Switch to Substances view, and instead of exporting the data, this time use the Reports feature to individually select data and add it to a report. After selecting relevant data, the report can be viewed, managed, and emailed after clicking the Report button. Thank you for watching. Please contact me if you have any questions.